I do want to introduce our keynote speaker, who is Mr. Tony Hall from the Royal Botanic Garden at Kew. Tony has spent his life in horticulture and his life dedicated to the study of Juno irises, which we have, at least during my tenure as editor of irises, we published several articles on Junos, because I think for the most part American gardeners don't really appreciate Juno irises as much as they could, so I thought, why not bring the world's foremost expert on Junos to our national convention to talk to us about his life's work in understanding Junos and talking a little bit about popularizing them maybe, or inspiring people to grow them in gardens. So with that, please give a warm American welcome to Mr. Tony Hall.
Thank you very much. At present, I recognize about 69 described taxa. Some are synonyms, so I'm not interested in those, and the main synonyms I've indicated next to slides. But there are about 12, up to 12 species yet to be described, and Arne Seisums, my Latvian colleague, and I will be doing that in the next few years. As you can see from the first slide, our informal groupings, which you'll see on the handout, including the main characters of the groupings, is based on a diverse number of science studies, and one of the most important of which is cytology. And it's worth mentioning that the cytology of Junos is very, very complex, with 2n equals 14 up to and including 2n equals 50. So a huge range of characters. And as with other iris species, like for instance the bearded irises, you get polyploidy, autopolyploidy, natural hybrids that have doubled their chromosomes and become stable new species, and disploidy, which is very important in Junos, especially where a population of a species either subtracts or adds 2 new chromosomes, either by fission or fusion. And when that happens, the same amount of DNA is present in the genes, so it's a good way of a species adapting to a changing environment and creating new species. We have autopolyploids, where a species doubles its chromosome and becomes a new species. And we have natural hybrids, which again then double their chromosomes so that they become fertile and stable and inhabit new territories. So that's the background to cytology in Junos. This is where I grow them, although it's not a particularly good slide. Most of the Junos at Kew are grown under the all-weather frame, which I designed and which was built in 1987. Sorry, can you all hear me? To put Junos in context, I don't agree with this ridiculous new paper that says genus Iris should be 24 or 25 new genera. As far as I'm concerned, they're all Iris. But at the bottom, you'll notice that the Junos are linked to the Nepalensis, that's Iris decora, etc., amongst all the bearded or crested irises. So even though they're bulbous, they are closely linked with the bearded irises. And subgenus Nepalensis has evolved to monsoon type of weather systems with a winter dormancy, whereas the Junos have evolved to a Mediterranean climate, so they're active or root active in the winter and completely dormant in the summer. But apart from that, they are closely related, and that is certainly justified by their pollen, which I'll show you in a moment. Parts of a Juno flower, I'm sure you're all familiar with it, but I'll talk about some characters, including flower parts, later on. The petaloid style lobes at the end of the style branches themselves, which are erect or ascending in Junos, the winged half of the falls. Some species are winged, some are not. and the blade of the falls, which is usually reflexed or held down to some extent, and the standards, which in most Junos are very reduced and held either at the horizontal or below the horizontal. So that's one of the main characters of Juno irises, apart from the fact that they have a bulb and fleshy or tuberous storage roots as well. And then, of course, with Junos, you have a pair of floral bracts, and the character of the bracts is often very important in Junos. Subgenus Scorpirus, as it's more correctly known, or the Junos, is divided into two sections by Rodionenko, which we accept, and section Juno consists of three distinct geographic areas: the Hindu-Kush alliance, mostly from Afghanistan, the Western alliance, so Western Asia and sub-Mediterranean, and then the Central Asian alliance, mostly Russian Central Asia, and then section Physocaulon. Physocaulon meaning swollen stem base, and this is a term that Rodionenko invented for those Curas irises that not only have a bulb and tuberous storage roots, but also have a swollen stem base from the previous year's flower as a third storage organ. So really interesting advanced development, but more on that later when we get to the Physocaulons. I've done extensive pollen studies of Junos and will be publishing a paper during the next 12 to 15 months. And Junos fall into three main types: football-like, I'll use that terminology here, which is found in the majority of Junos, where the plates on the surface of the pollen are in sort of hexagonal or polygonal shaped plates, anything from about 5 to about 25 or 30, or in a few species, hamburger-like, so only two plates, and the pollen grains are more subspherical rather than round. So that's a rare form. And then two species only have spiny pollen and what we call a winding aperture, where the pollen tube germinates. And both the hamburger and the spiny pollen have evolved from the football-like pollen. That's something we've learned with our extensive DNA and pollen studies. So the most ancient is definitely the football-like. But although Rodionenko considered the spiny pollen so different, he created a section, Acanthospora, for the two species with spiny pollen. That doesn't wash with me, because we find intermediates in some forms of Iris alcari between the spiny pollen and the football pollen. So clearly one has evolved from the other. But the interesting thing is that both the football and the spiny pollen also appear in subgenus Nepalensis, that is Iris decora and a few other species in that subgenus. And that indicates and supports strongly that they've evolved from a common ancestor somewhere in Eastern Asia. Section Juno, the Hindu-Kush alliance, 4 distantly related species, 2 with hamburger-like pollen and elongated flower stalks. What Brian Matthew described as a branching stem, but in fact, morphologically, it's an elongated flower stalk. So that's Iris cycloglossa and Iris aitchsonii, and then 2 distantly related species with football-like pollen, Microglossa and Stocksii. So cycloglossa, I've indicated the chromosome count, as you can see, and the geographic range as known of each species. So 2n equals 28, so a curious count straight away. Root system of cycloglossa, long and thin, and that's on the left, cycloglossa growing outside the old pyramidal alpine house, but here, accompanied by a fat dwarf on the left, is cycloglossa growing in the rock garden at Kew. And this is a wonderful species with a very strong carnation scent, growing to about 2 foot tall, and at Kew up to 9 flowers per stem. and it increases vegetatively very quickly and also from seed in about 6 years. Beautiful species, cycloglossa, referring to the round tongue-like hanging folds that you can see there. It was considered by Brian Matthew to be an ancient species, but our research and pollen studies have shown that it's actually a very isolated and quite advanced species, and it has a specialist large pollinator bee, only found in Herat province in Afghanistan. Aitchisonii in two color forms, like many bearded irises, either yellow or purple or blue. Afghanistan, Pakistan, and the blue form was discovered in Kashmir a few years ago on a herbarium specimen. There was a seed pod, so I nicked the seed from the herbarium specimen, grew it, and when it flowered, I was overjoyed to see it was the purple form of Aitchisonii, which had never been recorded in Kashmir before. So that extends its geographic range. As you can see, it's a rather spidery species with narrow leaves, but also the same kind of pollen. And you can see on the left how the hamburger-like pollen has evolved from the football-like pollen. So occasionally you get 3 plates, usually you get the 2 plates. It makes it more hamburger-like, but you can see it's evolved from a few plated Juno species into 3 and then to 2. And the rootstock of Aitchisonii is very characteristic, short, fat, and slightly squat. Microglossa is one of the rarer species from northeastern Afghanistan, very seldom cultivated and only collected a couple of times by Wendelboe and Gray Wilson and Hewer. So very rare in collections. And you can see quite clearly it has a very strong wing to the folds, characteristic of this whole group. And Iris stocksii, which we now grow very well at Kew from an Afghan collection, is the last of this group of irises, and in my opinion, Iris odontostyla, described a few years ago by Wendelboe and Matthew, is synonymous with stocksii. There's little to separate them except a slight difference in the shape of the folds. This is stocksii, wild in Afghanistan. This is odontostyla, or what was described as odontostyla type material, and as you can see, there's very little difference between the 2 species. The next part of section Juno is the Western Alliance, divided into 2 sub-alliances, Ochorei, Galatica, Stenophila, and Planifolia, and then sub-alliance B, Caucasica and Persica. Most authorities consider that Iris galatica and Iris stenophila are very closely related to Iris persica, but the DNA, morphology, and cytology has shown that that is far from the case, and Stenophila and Galatica are much more close to Iris ochorei in the first suballiance. Ochorei group, named after Ochor Eloi, who first collected material. Very characteristic, wide-winged, ruffled flowers, a slightly fragrant scent combined with cat's pee, so rather unfortunate combination. But still a very beautiful plant. And characteristic, apart from the wide wings, is that the leaf bases and bracts are slightly inflated and pale. That's a selection, lilac from Leylak Station in Turkey. Ochorei is widespread, and as it's widespread, it's very variable in color and in height. A good purple form and snowflake, a good albino. Species nova with the same chromosome count is from just north of Palmyra in an isolated mountain range. It's like a very squat form of Ochorei, but it has very tiny seeds and very grey foliage, and that's something we've yet to describe. So though it's the same chromosome count as Ochorei, it's quite isolated geographically, and it needs a name. And then in the Munzer range of Central Anatolia, there's another which is probably a new species because it has a different chromosome count to Okori, and this is at the moment known as Iris af Okori Munzer rebel, named after the Munzer range. And the next slide is dreadfully out of focus, but I stole it off of the internet and I couldn't get a sharper image, but that shows you growing in the Munzer range and in the valley at the foot of the range, and I have never ever seen Junos growing so thickly like that as a carpet. It's wonderful. And there's also Iris fumosa, which is found only northwest and west of Aleppo in Syria, and that also is 2n=24, so different to Iris okori. And both have rhomboid rather than paddle-like standards, and as I said, they have a similar count of 2n=24, so they will not hybridize and produce offspring with Iris okori, so they are separate elements, separate species. Iris peshmeniana I described about 5 years ago with Adel Gumer, and this is from Malatya province, and an isolated white and yellow species. In some ways it looks a little like Iris nusariensis. It's very short, very squat, with very inflated pale leaf bases, but geographically they're hundreds of kilometers apart. And there's nusariensis itself with the same chromosome count and endemic to the Jebo-Nusari range, hence nusariensis. There it is, always in the blue range. And Regis Uzziah, named after King Uzziah from the central Negev, reigned that part of Israel centuries ago. Blue color range, and then related to it is Af Regis Uzziah from southern Jordan, mostly the Edom and Doab region. Again, a different chromosome count to Regis Uzziah in the narrow sense, so they're separate species. And Dinsmore, who collected material in the Doab region, was going to call it Doabensis, which is a good name, but he never validly published it, so we'll probably end up using his name and call it Doabensis. It's much more variable and larger than Iris Regis Uzziah itself. Galatica, only a single species, of course coming from Galatia in Turkey originally. As in Iris stenophila, you can see the junction of haft and blade to the falls is always pinched, as if between finger and thumb, and that's never the case in Iris persica. And Iris galatica is always 2n=24. A tiny species, but variable in color. Again, you get yellow forms and purple forms, and bicolored or veined forms. Widely winged, but the most important thing is that the bracts of Iris galatica, as you can see on the left, are both more or less green. In Iris persica, they're not. The stenophila group, stenophylla meaning narrow-leaved, Iris stenophila in the strict sense is a very beautiful, large plant from the Cilician Taurus with huge flowers and beautifully scented. Quite variable, but always has narrow silvery foliage which is long attenuate, so gradually narrowing. Aphstenophila, described originally as Iris tyri, and we in fact will resurrect that name, is mainly the Sicilian Taurus, very variable. Chromosome counts vary, so disploidy is certainly occurring in Stenophila. So tyri is very variable, but always small. And then Stenophila subspecies alisonii, which we will eventually call Iris alisonii because it's very distinct, is always in the blue range with the heavily veined blade to the falls, and always 2n equals 26. And that's the extreme western Taurus, so geographically quite isolated, but a beautiful little species, not so easy to maintain. And on the eastern end of the Taurus is another undescribed species, again very variable, usually with both bracts slightly translucent or very soft textured. Extremely variable, but often very muddily coloured. But it's been collected a lot in recent years, but has yet to be described. The Planifolia group, or Acanthospora, as Rodionenko described them, because of the strange spiny pollen. The only Juno species that goes into Europe, so Spain, Portugal, a number of Mediterranean islands, Sicily, and North Africa. Always in the blue-purple range, often nicely veined, as you can see. Sometimes huge plants, up to a foot tall. Rarely you get albinos, and even more rarely on the right from North Africa, a pink form, and pink is very, very unusual colour in Junos. Related to Planifolia and with the same chromosome count is Iris palestina. There are steps to give it subspecific rank, but I prefer to keep them separate because geographically they're very different. It's a smaller, weaker plant with much more range in colour, so pale blue, pale green, yellow, white, and much more difficult to grow. But there are some lovely colour forms in the wild, including a sort of azure blue form, but never the purple blue that you see in Iris planifolia. Planifolia, incidentally, is probably the oldest cultivated Juno, cultivated since the 14th century and certainly described as early as that, so earlier even than Iris persica. The Western Alliance, Suballiance B, Caucasica group from the Caucasus, of course, and then Persica group proper. Caucasica group, characteristically inflated and keeled bracts, and although the flowers are widely winged, the blade of the falls is also wide, so there's very little differential between the blade and the wing in the Caucasica group. Subspecies Caucasica, mostly Caucasus, but also just extending into northeastern Turkey. And then Caucasica subspecies turcica itself, which is extensive throughout central and eastern Turkey and also into northeastern Iraq, where it's been collected, I think, a couple of times. Always yellow, but occasionally with blue in them. And then recently described Iris nezahatiae from a tiny area in northeastern Turkey. It's closely related to Iris caucasica turcica, but it's really just a colour form. It has the same chromosome count, so again, we will probably reduce that to either subspecies or varietal status when we produce our monograph. The Persica group, very complex, and what was originally in old illustrations from the 18th, 17th, even 16th century as Iris persica is in fact Iris stenophila sensu stricta, including all of these old plates. So we are going, because Linnaeus never produced a type specimen of Persica, we are going to have to redescribe and retypify Iris persica if we want to conserve the name. And we do want to conserve the name because it's the type species of the whole of the Juno group. And within Iris persica itself, as we understand it today, there are a number of discrete geographical entities that are pretty stable. One is Barmardinensis, but 2n equals 18 to 20, though disploidy has occurred here. And you'll notice that the inner bract is always papery and translucent in Iris persica, and the outer bract is erect and green-like. Very subtle but important character. Variable, but usually in Martinensis, the flower tepals are edged in silver, which is really beautiful. Difficult species to maintain, though. The northern element of Persica, it comes from an area where Galatica is more common, and I think it has evolved to the same pollinator, because you get yellow and purple forms as you do in Galatica, and the differences between the bracts in the northern element are not always obvious until after flowering. So this is all the northern element, but with the same chromosome count of 2n=20, and then the southern element from desert areas of Turkey and northern Syria, where the blade of the folds doesn't have the prominent patch that you see on most parts of Persica. And then a Manus element from the Amanus range in southern Turkey, just above Syria, which is quite distinctive. But the most distinctive, which was once described as Iris ceana, so we will probably resurrect that name, is the western element. So throughout the Taurus range, really widespread and variable, but the flowers generally are sort of monochrome, and they have a prominent white patch either side of the raised yellow crest, and the standards are, in comparison to the length of the folds, really quite short, but very variable, and the easiest form of Iris persica to grow. Wallisii, which we described just about 4 or 5 years ago, named for Randwick Wallace, who first found it in Mount Hermon, is related to Persica and has been mistaken for Persica by the local botanists, but the two bracts in Wallisii are semi-translucent, which they aren't in Persica, and it has a distinct chromosome count. So an interesting, although not particularly attractive species. Hymenospaitha is the relative from Iran. Iris persica has never ever been found in Iran, but in those days Persia was a much vaguer geographic name than it is now. So it probably included parts of other countries at the time. Hymenospaitha, subspecies Hymenospaitha, both bracts are completely translucent and silvery. And it also has long tapering silvery foliage, but difficult species to grow because it's a real semi-desert species from southern Iran. Related is subspecies Leptonora, which really means not silvery veined, normal veined most of the time, and it has shorter, more abruptly tapering foliage, but apart from that it's very similar, except its chromosome count is very, very different, so we will probably give those two species separate species status. Pseudocaucasica, which is complex and we're just beginning to understand it. 2n=22, with a very limited distribution, and after flowering their internodes are visible in the stem, so the stem elongates slightly when the plant is in fruit. There it is in its type locality in Azerbaijan. Koyankui has just been described by a Turkish botanist, but he won't let me have material, so I can't study it. And so whether it's related to Pseudocaucasica in the strict sense or Pseudocaucasica, I don't know yet. But it has internodes at flowering time, but it's very robust, so we're not sure what to make of it. Pseudocaucasica is always compact, even in fruit. And 2n equals 20 usually. And this is in Turkey, adjacent Iran, and also Armenia and Azerbaijan, so it's extremely variable, but always very, very squat, as you can see. Variable in color, and there it is from Armenia and Azerbaijan, with 2n equals 18 to 20, so disploidy again is occurring in this undescribed species. It is often, the Armenian form has often been described by present-day botanists as Atropatana, but there's nothing to do with Atropatana. True Iris Atropatana is a synonym of Iris caucasica. One of my favorite little Junos from the Edom region of Jordan is Edomensis, which always smells of boiled sweets and has a very characteristically shaped flower. Beautiful, very difficult to grow, but absolutely lovely. Rare, restricted endemic of Jordan. And then Iris postii, the most challenging Juno to grow, a collected bulb, collected for the flora of Saudi Arabia by a botanist. Took me 10 years to flower from a collected bulb, and then it flowered every 3 years before it finally died, sadly, but we were able to study it and paint it and do the chromosome and pollen studies on it before. It's very widespread in the Syrian desert, but very, very scattered, and this is the only time it's ever been successfully cultivated outside of Israel. It's a strange thing because superficially it looks like Iris ochrae. It has inflated leaf bases and internodes visible sometimes during flowering, and a flower shape rather like Iris ochrae. So when the initial DNA results said no, it's more related to Iris persica, I said I don't believe you. So we put a second material from another part of its range into the DNA matrix, and it came out with exactly the same result. So obviously convergent evolution is occurring as it does with a lot of characters in genus Iris. So it just happens to be coincidence that it looks superficially like it should be a member of the ochrae group. But that was a surprising result. In the Persica group, you have Iris fosteriana, which is a bit of an oddball because it's the only one I've shown you so far which doesn't have wings to its folds. It also has these very long, thin bulbs and very wiry, fragile roots, and is a challenge to grow, named for Sir Michael Foster, of course. And very clump forming, as you can see here in the wild. And it's a curious thing phytogeographically because it sits in the bottleneck between the western species of Junos and the Central Asian species of Junos, and it reaches both sides of this bottleneck, and most bulbous species don't do that. They're either one side or the other. So although without having winged folds, it looks much more like a Central Asian species, in terms of evolution, it's more related to the Western Asian species. So a fascinating species. Central Asian alliance, the largest alliance, is naturally divided geographically into the Pamir Alai group and the Tian Shan group. So very natural division. Two species in the Pamir alliance extend to the Tian Shan though, and the Pamir suballiance is a group of species without wings, Bucarica and its closely related species, and with wings, the Maricandica group, and you'll know Iris magnifica from the Maricandica group. Bucarica sensu stricto, 2n equals 22, widespread in Tajikistan and Uzbekistan, always bicolored, so white or pale yellow with a darker yellow blade to the folds, vigorous, easy outside. Cultivated Iris bucarica, which used to be known as Orchioides, in the 19th century, but Orchioides is an entirely separate species. Related to Iris bucarica, but we have yet to describe it, with 2n equals 20 to 22, so disploidy occurring again is in northeastern Afghanistan and adjacent southeastern Tajikistan, so geographically a little separate from bucarica in the narrow sense. Dwarfer plants on the whole, and usually monochrome, yellow or pale yellow or creamy white. And then related to bucarica is vicaria, with the same chromosome count, 2n equals 22, and always with violet or purple stained flowers, and as you can see, internodes visible during flowering, and equally easy to grow outside if you can get hold of vicaria. Vicaria is interesting because with 2n equals 46, and originally we thought it was an autopolyploid, that is, vicaria had doubled its chromosome count, but our study of its cytology and karyotype, that is the shape of the chromosomes, indicate that it's a hybrid, a natural hybrid between Iris bucarica and Iris vicaria in the narrow sense, and then it doubled its chromosome count and became stable and fertile, so an autopolyploid. A very different chromosome count because vicaria itself was 2n equals 22, and this, with double the chromosome count and then two extras, 2n equals 46. So it's a species that has evolved to fill an entirely separate niche. And usually it's smaller or dwarfer. Flowers are white with just a hint of purple staining on them. But apart from that, morphologically, they look very, very similar. And here's a lovely patch of vicaria cultivated by my old friend Panayoti Kalaitis in Denver, where I'm going this weekend. Clump forming, as you can see. And related is Iris worliensis, 2n equals 24. Quite widespread in Uzbekistan and Turkmenistan, but again it's a species that's derived from a natural hybrid between Iris vicaria sensu stricto, that is the very coloured one, and Iris narbutii, a very dwarf species with a dark blade to the folds. And they both have the same chromosome count, but then a couple of chromosomes have been added on with disploidy, and it has extended through its own range. Quite variable, very characteristic with this dark blade to the folds and bright yellow patch around the crest. Parvula group, parvula because they're rather small, delicate plants. Parvula itself, very distinct, with long, attenuate, narrowing, silvery foliage, and the blade of folds with this lovely dissected crest. Not the most horticulturally beautiful thing, but Vedenskyii, which is on the left in this slide, is synonymous with parvula, the same count, everything about it is correct. And then Casanovii, recently described, very long attenuate foliage. Again, it can have a very fimbriate crest and a completely different chromosome count. So my Russian colleagues are quite correct in giving this species status. It's endemic to the Turkestan range. Very, very variable. In the past, this was grown in England as parvula, but it's a very different species. Related is Linifolia and Linifolia formis, if you like, a yellow-flowered version of parvula or Casanovii. And it has a strangely disjunct distribution, that is, it occurs separately in 3 isolated areas, but it's all the same species. Again, a very small species. And then Tadchicorum, little is known about it except that it occurs in the Darwas range At the moment, I don't know its chromosome count. I did grow it, and this is a picture of it at Kew, but it died before I was able to molest its roots for a chromosome count. But friends have collected new material last autumn, so hopefully I'll improve our knowledge about it soon. And also in the Parvula group, but it's in its own subgroup, is Iris narinensis from the Narin River Valley of Uzbekistan and Kyrgyzstan. So, if you like, halfway between the Tian Shan and the Pamir-Alai ranges. Again, very fimbriate crest, and superficially it looks much more like a member of the Kushkovitziai group, which I'll come on to later. And like all of the Parvula group, it has a tiny, short-lived arrow to its seed, which is the seed stalk, of course, the arrow. But sometimes it drops off when the seed matures. Interesting little species. And then the Narbutii group, I mentioned Iris narbutii as being one of the parents of Iris worliensis, the natural hybrids. And Narbutii, including Iris rudolphii, is from the Pamir-Alai and the western Tian Shan. So, like Iris linifolia, 3 distinct areas of distribution. One of the main characters of Narbutii are these huge paddle-like standards, which are usually very colorful as well, although in albino forms the color is not pronounced, as you can see on the right here, but a beautiful little species. Narbut was a Russian commander who led expeditions in parts of Russian Central Asia in the 19th century, so it was named for him. And Rodionenkoi, recently described, named after Georgy Rodionenko, of course, related to Iris narbutii, but without the large paddle-shaped standards. The standards here are 3-lobed, as you can see. And again, the Russians have not been able to let me have material to play with, so I can't say much more about it at the moment. The Maricandica group, very wide wings, strong, robust stems with internodes showing, and quite thick storage roots that taper abruptly, and 3 very closely related species with all the same chromosome count, and in the wild they do sometimes hybridize. Maricandica white, it's a sort of pale, creamy, translucent, as you can see, very widely winged. Related is Iris svetlanae, which Brian Matthew did consider synonymous with Maricandica, but we've shown that it deserves species status. And in a stupid recent paper by a couple of really ignorant Russians, I get on with some Russians enormously, but there are some people who publish new names and do not understand the material they're studying, and Victoris and Petri, as far as I'm concerned, are synonymous with widespread and variable Iris svetlanae. Widely winged, translucent, yellow flowers. Victoris was described because it has a bilobe standard, which is a slight local mutation and not enough to give species status. Crazy, absolutely crazy. And Petrii with broad wings, but then Iris svetlanae has broad wings, blunt tip standards, which most svetlanae have, and arching leaves, which, as you've seen from the previous images, Iris svetlanae has. So sometimes I lose my patience with these botanists. Hippolyte is the 3rd of the Maricandica group, and one of my favorites. It was thought to be endemic to Mankochka, this little hill in the Kizilkum Desert, but we've also found it in the Nuratau range. So it's like a blue or violet version of Svetlanae or Maricandica, if you like. A beautiful thing. Hippolyte is a Latinized version of Impolite, and Ippolit Granitov was the Russian botanist who first discovered it. So it's named after Ippolit Hippolyte, so nothing to do with horses. But a very characteristic, beautiful species, but again, it does integrate with Maricandica in nature occasionally. And also in the Maricandica group, but separate, is Iris magnifica, so it stands out as an isolated species, as you know, very easy in the open garden, up to 1 meter tall, and this is the thing that gave Juno's the misnomer, if you like, of leek-like or sweet corn-like, because of the arrangement of stem and leaves. A bit of an insult for Iris magnifica, which is a beautiful scented thing. And as you can see from the pollen, it's one of the species that has the fewest number of plates. Plates in Juno's, in the football type, range from 5 to 30, sometimes even 35 plates. And magnifica has the smallest number of most of them. Magnifica virginity or Magnifica alba, and the rootstock of Magnifica is very characteristic. Long, thick, finger-like roots and relatively small bulbs, so most of the energy is stored in these very deep rooting storage roots. And Iris Magnifica Margaret Matthew, which is a collection that Brian made with lovely edges to the falls, I named for Brian's wife, Margaret. Brian is still alive and well, even though he's long retired from Kew. The Tian Shan suballiance, wholly Tian Shan in distribution, so a geographically very stable group. The Orchioides group, which has wings to its falls, and then the Alba marginata group, the Alba marginata subgroup itself with wings, and the Kushikavitsii subgroup without wings. Orchioides group, named because the original specimen looks a little like an orchid, according to Carrier, the French botanist. 2n equals 30, very widespread, very, very variable, always widely winged, lovely translucent flowers in the yellow range, and this wonderful punk rock swept back, highly dissected crest. A really lovely species, sadly unscented. If it had a scent as well, I'd fall in love with it really. But here you can see it. It's such a gorgeous, gorgeous species. Orchioides. Related to Orchioides, and which in a big article on cultivation of Junos some 15 or 20 years ago, I misidentified as Iris tubergoniana. But as you can see, it has internodes, and it has not a wide wing, but a fairly wide wing, and it has very, very fleshy, almost magnifica-like roots. It has the same chromosome count as Orchioides, but it has a number of features which separate it quite distinctly. One is the unusually long, tapering upper leaf, the internodes, which are visible at anthesis, and the very wide wings to the falls. So it looks a little like tubergoniana, but it isn't tubergoniana. And it smells of boiled sweets mostly, so rather nice. And then true tubergoniana doesn't have a very wide wing to the falls, but has a nicely dissected crest, but never has internodes showing during flowering. Here's a picture of it in the wild. As you can see, there are no internodes showing. It's always this bright canary yellow, a lovely little species, tubergoniana. And related is Capnoides, widely winged again, but three-pronged foliage, internodes showing usually in the blue or violet range, and recently described and a valid new species in its own right, dwarfer paler, is, is Ostrocheilus Chatkalica, a bit of a mouthful, but it just means from the southern part of the Chatkal range. It has the same chromosome count, but a different karyotype, that is chromosome shape. And its seeds do not have the prominent arrow that Iris capnoides has. And then there's Iris pseudocapnoides that Yanis Ruxans described a few years ago, which is a bit like capnoides but taller and without a heavily dissected crest, and always pale white or straw yellow, vigorous and fairly easy in the open garden pseudocapnoides. The Alba marginata group, Alba marginata itself is a tiny little thing and one of the most difficult Russian species to grow because it comes from long, hard, cold winters, and in Britain it must have a long, cold, hard winter to flower. This year, thankfully, I managed to flower it and even got a pod, so now I can study its seeds and everything else about it. This is true Alba marginata, as you can see, widely winged and a nicely dissected crest, and always in the pale blue, violet, heavily veined range. Related is Iris graberiana. Now, when Ernest Seism and Yanis Ruxans collected Iris zenidae from its type locality in Kyrgyzstan, they sent me or brought me material, and as soon as I looked at it in flower, I said, Iris zenidae is synonymous with Iris graberiana. And Ernest said, are you sure? I said, well, I'll do a chromosome count because I know that Iris graberiana has 2n equals 50, the highest count of any Juno species ever recorded. We did, and it has 2n equals 50. And there's no doubt that Iris zenidae and Iris graberiana are one and the same species because this collected material looks identical to the type material of graberiana and Q. cerberium. Graber was Van Tubergen's main bulb collector at the end of the 19th century and beginning of the 20th, and he collected in Turkestan, which is a rather vague term for Central Asia. So unfortunately they didn't know the type locality of graberiana, but now we do because it's the same as Iris zenidae, and the Russians have finally accepted that Iris zenidae is in fact synonymous with graberiana. 2n equals 50 is because Iris graberiana is an autopolyploid of Iris alba marginata. So it doubled its chromosome number, and then dysploidy occurred, and it became a fertile autopolyploid inhabiting a completely different area. And of course, as an autopolyploid, it's much bigger in all its parts. So that's the origin of Iris graberiana as a natural autopolyploid of alba marginata. Fairly variable. These are a number of selections collected by Ernest and Yanis from the type locality. And Wilmotiana is the third member of the alba marginata group. Very common in cultivation, characteristically blue or pale blue with a big white patch on the blade of the falls. Lovely thing and fairly easy outside at Kew, so one of the 5 or 6 species that we can grow outside. It's robust but compact at the same time, as you can see, and has a very characteristic, fairly long, narrow rootstock, and sometimes the wings to the falls, as you can see in this image on the left, are not well expressed. So in fact it falls halfway between the alba marginata group and the next group, the Kushikavitsii subgroup, which has long wiry roots like Wilmotiana, and its flowers are generally unwinged or have the slight hint of a wing. Kushikavitsii is widespread, but I must say here that this is one of the groups and the only Russian group of Junos, I do not fully understand, because one needs to study all populations in the wild, in the whole of Russia, to really understand what's going on. All I can say is I'm not convinced that there are four separate species. Kushikovitsii is usually very easy to recognize because it has an ink blue blotch to the blade of falls. And then Alma atensis from around the city of Almaty in southern Kazakhstan, but northwest of Almaty and southeast of Almaty you have pure Kushikovitsii, and Alma atensis is greenish flowered, and some Russian authorities place it with Iris subdecolorata, but as far as I'm concerned it could just as easily be a greenish flowered form of Kushikovitsii. Now, the literature states that Alma atensis is probably extinct in its type locality, but in gorges next to the type locality you get green-flowered Kushikovitsii, or what the local botanists are describing as Kushikovitsii. So I think there's a lot in what I'm thinking about this, that it's closer to Kushikovitsii. Here you can see a yellowish or brownish blade to the folds, but apart from that they're very similar, and all of the species of this group have the same chromosome count. And then you've got Iris inconspicua, which is said to have a not very prominent markings on the blade to the folds, but the markings can be so variable anyway. And subdecolorata, pale flowered, but again there's not much difference between that and Kushikovitsii as far as I'm concerned, so I'm a little confused by the literature on this and the images one sees on the internet. So I better not comment more. The final group is the Physocaulin group, or Rodianenko's section Physocaulin. He divided into the Drepanophyllae series and the Rosenbachianae series, but Drepanophyllae is invalid as a series name because it has to be named after the section, so we're calling it Physocaulin group, and then we're accepting Rosenbachiana but calling it the Rosenbachiana group. But the species he placed in each section we completely disagree with, so we've regrouped them. Characteristic of Physocaulin section is a very prominent aril running often the whole length of the seed, as you can see on the left here, and the aril is a modified seed stalk. And as I mentioned at the beginning of the lecture, Physocaulin means swollen stem base, and the central arrow on the right indicates last year's stem, which is swollen and stored all the food from the previous year's growth, and on the side you have smaller true bulbs, and then characteristically of the Physocaulin group you have these huge returned, or it was returned and housed somewhere where it shouldn't be. And as there are millions of slides at Kew, there's no way I'm going to go through all their slide collection to find it. But it's very closely related to the two previous species, Versiorum and Zaphropogeva dewey, but it has much longer attenuate bracts. And that's all I can say about Cabolica. There's another image of it, as you can see, downturned folds, no wings, very grey-blue foliage, which you can rub off with your fingers. And lastly, that's me, of course. I'm sure you recognize me straight away. And although, yeah, DNA work and all these modern tools are useful, the best diagnostic tool is always your eyes. So if you grow the plants, you get a real feel for them, and you look at their rootstock when they're dormant, the way they grow, and everything about them. So eyes are still the most important thing in Junos. Thank you very much.